I'm not sure I want tapeworms to be plotting together. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klinowski, your host for today's episode. Stem cells have been in the news lately due to their potential to create new medical treatments. But stem cells aren't unique to humans. Tapeworms, a longtime scourge of humanity, can use them to regrow lost portions of their bodies.
Our guest today is Dr. Tania Rozario, assistant professor in the Department of Genetics at UGA and a member of the Center for Tropical and Emerging Global Diseases. Dr. Rozario's research concerns stem cells and the signals that regulate regeneration in flatworms. Tania, welcome to the podcast.
Thank you so much for having me.
So, Tania, one thing we like to ask our guests, usually as their first question, is to relate their path into scientific research. What first interested you in science and eventually interested you in developmental biology?
I've just always been fascinated by the amazing diversity of biology around us. I mean, we as scientists work hard, but we cannot even remotely compete with evolution in the number of experiments that it has already done for us. And it just behooves us to try and understand just how nature can fix so many problems, how so much diversity in biology can occur. So to me, actually, my interest in science was super basic. I think there are many different paths that I could have taken. And I just took the paths where I thought that there was so many open questions that I could answer, so much to learn, and people that I really wanted to join up and make a community of scientists with.
So on the genetics department website, they list you as a developmental biologist.
I am.
But your work really covers a lot of other disciplines, such as parasitology, genetics, and stem cell biology. So what are the challenges of this kind of multifaceted research?
It is challenging sometimes. You don't really fit into one pigeonhole. But that's also a plus. People talk about interdisciplinary research all the time. And sometimes, that concept only exists because you imagined boundaries around your discipline to begin with. And I feel like I've been able to navigate through this wonderful mesh of parasitology, basic developmental biology, genetics, molecular biology, biochemistry, without much of a problem, because at the end of the day, everyone is just motivated by answering questions. So I think I've been fortunate as well that the communities that I have been working with are also just open to big umbrellas. Parasitology has everything from tiny microbes, right, to giant tapeworms like mine. And developmental biology also, from the earliest mechanisms that actually result in the oocytes in sperm to post-adult development also, even processes like regeneration, which typically we're talking about in an adult context. So I think all of these disciplines are vast, and there's a lot of opportunity for people to come in with different perspectives, different histories, different techniques, and still make meaningful discoveries and still generate excitement from others who maybe don't traditionally follow your kind of techniques or your kind of path.
That's a really good point. Even just thinking of parasitology, because scientists, we love to categorize things, but in even just parasitology, we have immunologists, we have cell biologists, we have molecular biologists, we have biochemists. So in a way, we're all a bit multifaceted, but I think yours stands out.
I think so, too. And I think it's great that you embrace it and don't try to put yourself in a box like they often ask of us on forums. And
Yes.
to describe ourselves. We can only usually choose one.
Yeah.
But a lot of us really cover more than those things. So, Tania, let's start talking about your research here.
Okay.
And you mentioned the Regeneration Project, and we also hinted at stem cells in the intro. So why don't we start there and start by talking about stem cells, and maybe you can integrate your research into the conversation. But before we get there, we really want to start with the basics. So what are stem cells?
Stem cells are cells that really do two things. One, they have to self-renew. They have to make more of themselves. And two, they have to have the capacity to differentiate into at least one other cell type. Sometimes they can differentiate into many cell types, like our hematopoietic stem cells that can make all of the white blood cells and red blood cells. Sometimes they're only going to make one cell type, like spermatogonial stem cells that are really only going to make sperm. And then you have those magical stem cells that we refer to as totipotent or pluripotent, where they can make all the cell types that are needed to sustain the adult organism. And those are the kinds of stem cells that are most relevant to certain kinds of regenerating organisms that actually retain these resident pluripotent stem cells that they can then deploy later in life and actually make all of the tissues that the adult needs to survive. And stem cells have been fascinating to me because they seem like magic. But across the plant and animal kingdom, there have been so many different strategies to try and access this state where you can essentially go back in time, reset the cell so that now it can make a cell type that is needed for the purpose of the time that it is in. And in animals that can regenerate their whole bodies, of course, they must be able to make all of the adult stem cells. And so that started to become more and more fascinating to me, and that is what drew me to the flatworm phylum, which is full of worms. Many of them are actually free-living. And then you have this one monophyletic clade that is exclusively made up of parasites. And across this phylum, there's actually a lot of different regenerative ability. You have the Ferraris of regeneration, which are freshwater planarians like Schmittea mediterranea, and they will seemingly regenerate from almost any fragment that you cut from this worm. If you take this worm and you cut it into 10 pieces, you will get back 10 worms.
Wow.
And then there are, other worms, for example, schistosomes, which are parasites that infect humans. They don't regenerate at all. So, people have tried to cut them into half and they don't ever regenerate their whole bodies. But really throughout the flatworm phylum, you have everything in between. Parasites that can regenerate from some amputations only. Maybe they can make their heads, but they never make their tails, or the other way around. Or they only regenerate in certain contexts. So clearly there's a lot of diversity of mechanisms that regulate how and when and in what context these stem cells operate. And so that's what drew me to the phylum, to try and understand not just the signals that promote regeneration, but also those that restrict regeneration, and how much we can learn from that, and how then we can apply that to the parasites in the clade, including tapeworms.
So let's say you've got a flatworm in the lab. How do you know which cells in there are the stem cells? How do you identify them?
Right. That is actually a challenge. At a basic operational level, they have to be dividing, and then their progeny will need to give rise to now a differentiated cell, say a skin cell or a muscle cell. So in some ways it's difficult because you need to be seeing this live, but most of the time we can't do that. So we interpret that by labeling dividing cells and then labeling their progeny with markers that we have previously established and associated with different tissue types, using methods like in situ hybridization in order to stain the RNAs that are, what is the word I'm looking for? The RNAs that define a certain cell type. Because in the end of the day, all of our cells have exactly the same DNA, but what makes them different is the RNAs that they express, and therefore the proteins that are operating in those cells.
So your work suggests that stem cells take cues from other cells around them.
They absolutely do. And that is not unique. That is not some unique thing that I came up with. Mm-hmm. This is universal for stem cells. All stem cells need friends. They're getting their cues from their environment.
So what are some of these cues?
So a lot of them are secreted factors that are made from cells that define a stem cell niche that is actually necessary to promote stem cell behaviors, for example, their capacity to divide or their survival or even their differentiation potential. So they can be things like secreted ligands like WNTs or FGFs or BMPs. These are all major signaling components that have been characterized in many different stem cell systems. But they can also be things that maybe we don't think about as often, like amino acids, or even non-traditional modified amino acids, or little amino acid chains that can also act as signaling molecules. They can be secondary messages, of course. It can be almost anything. And a lot of the times we're looking at signals that are right there adjacent to the cells. But it's also possible that stem cells can respond to much more long-range cues that even come from other animals, for example, other animals in the environment, maybe allowing the tapeworms to actually talk to each other and sense how many different organisms there are in the intestine at any one given time.
I'm not sure I want tapeworms to be plotting together.
It sounds like a real horror movie. But again, another horror movie that hasn't been made yet.
So I had one other question about stem cells, and oftentimes we talk about markers on cells, and that can often be proteins on their surface, or as you've mentioned, RNAs that they express. Are there any stem cell markers that are actually kind of conserved across organisms?
There are. A lot of stem cell markers that are conserved across organisms are the things that the stem cells need in order to divide. So the dividing cells are going to need to replicate their DNA. They're going to need to package that DNA into nucleosomes. They're going to need to undergo the cell cycle. So all of these things have been well characterized as dividing cell markers. So the difficulty becomes that in many systems, it's not just the stem cells that divide. They also have progenitors that are also capable of dividing, but they have lost their capacity to be stem cells now. In the flatworm system, all the dividing cells are stem cells. They divide and they make progeny, and they don't have stores of dividing progenitors. At least that is the dogma. And to be perfectly honest with you, I am suspicious of this dogma for tapeworms. And that is one of the questions that we need to resolve here, whether or not there is actually more quiescent populations or slow cycling populations of stem cells within the tapeworms that then generate these dividing progenitor populations that have actually lost their stem properties. So that's a long-winded way to say that it's not that easy to define a stem cell just by the genes that they express. At the same time, we have found many conserved genes that are redeployed over and over again in many different whole animal regenerators. One really great example is the gene Peewee. Now, this is a guardian of the genome. So you can imagine that when you have stem cells that are going to give rise to all the adult stem cells, it is really important to protect against jumping elements within the genome that are going to create mutations that you don't want to carry on to all of the progenitor cells. So Peewee, for example, has been a well-characterized and highly conserved gene in many pluripotent stem cell systems, from planarians to hydra to ACLs, a lot of different organisms. But they are absent in the genomes of all the parasitic flatworms. And there are actually a number of well-characterized conserved stem cell regulators in free-living flatworms that are absent in the genomes of all parasitic flatworms. So clearly there are alternative ways that the parasites have evolved to deal with some of the same challenges. And so that is another big motivation for why I am interested in pursuing tapeworms and not just relying on what we can learn from their free-living cousins.
So I think we'll jump to the tapeworm model in a second, because I think that's really important that we speak to why it's such an important model. But before we do that, I just want to ask you one question, because I think our listeners might be interested in this. We're talking about stem cells in worms, but can we learn anything about regeneration in humans by studying tapeworms?
I hope so. I mean, ultimately, we're all made of the same stuff. So I always think of it as a twofer situation in my lab. Either we're going to learn stuff that is conserved, that can be applied to many different systems, including hopefully to humans, or we're going to learn about some parasite-specific strategies or the way that parasites use their own unique genes that maybe don't share homology to other animals, including humans, and that will allow us to design strategies to actually combat those parasites directly.
So you've touched on the fact that you use the rat tapeworm in your research and some of the advantages there, but let's cover again what makes them a better model system than the free-living flatworms, because they've got to be more difficult to maintain in the lab.
They are more difficult to maintain in the lab, but one of the reasons that I chose this rat tapeworm model is that they're actually very simple to maintain in the lab compared to other tapeworms, which would be really very, very difficult. And people do it. There's some wonderful work being done with a variety of tapeworm species across the world, including a really great working group in Peru that is trying to really understand the pork tapeworm, which is the most dangerous tapeworm to human beings. But take the pork tapeworm. If I wanted to establish a molecular biology research system in which I had to maintain this tapeworm, I would need pigs and human beings in order to complete this life cycle. Whereas I need beetles and rats to complete the life cycle of Hymenalepis diminuta, the model system that I work with. But as I said, there's just going to be many differences between the free-living worms and the parasites that make it important that we don't just make assumptions that everything is going to be comparable. So I've given the example of how there are these conserved genes in stem cells that are not even there in the genomes of our parasites, but they also have all kinds of different biology. One of the features that unites all of the parasitic members of the clade is their very special skin. So the name of the clade is the neodermata, for new skin, because their skin is nothing you have imagined before. It is not an epithelium at all. It is one cell. There is one cytoplasm that covers the entire worm, and there are thousands of these cytons that carry the nuclei that make small cytoplasmic bridges to this cytoplasm that is shared around throughout the whole worm. So if I were to show you a rat tapeworm and it gets to like 40 centimeters or a meter long, that is one cell that is covering the entire body of the worm. This is true for the whale tapeworm that could be 40 feet long.
40 feet long for a whale tapeworm.
Wow.
Oh, my goodness.
This cell sounds like a giant Snuggie to me, too. Just wrapping around and keeping it all cozy.
So there's all kinds of cell biology that's quite different within the parasitic flatworms as well. A lot of their tissues are syncytial, and we haven't even begun to really understand how those cells are made and formed and maintained, and how that would differ with other free-living flatworms that do not have the same kind of anatomy.
Can you talk about the syncytia for a second, just for listeners, so they understand what we're talking about?
What is a syncytia?
Yeah.
Right. So, a syncytia is just a tissue that is made up of multiple nuclei, but not delineated by cell bodies. So there are many different ways that you could do this. Like we have multinucleated muscle, for example. You can have muscle fusion going on so that you get these long muscle fibers that have many different nuclei in them. Or you can have this crazy situation like I just described, where you have one part of the cell that is shared, like the cytoplasm, and then these cytons that contain the nuclei, which have all the DNA, that are kind of individual. You can imagine them as like beads on a string, really, that are all connected by a little connection.
It's fascinating.
So in addition to being very productive at regenerating themselves, the tapeworms are equally productive at replicating themselves. Can you tell our listeners about that?
Yes, they are baby-making machines. So if you think about the anatomy of a tapeworm, and there is a lot of diversity, but most of the tapeworms that infect humans, for example, in their adult stage have a head that has some kind of organ that allows them to attach to the lining of the intestine. Behind in many of these systems, we have an unsegmented part of the worm that in my lab we call the neck. It's sometimes called the germinative region. And in the rat tapeworm, this can be as small as a millimeter. But then you make the entire body of the worm, right? So now we're talking about something that could be centimeters or meters long. That is made up of thousands and thousands of segments that have a very unfortunate name. They're called proglottids. And each and every one of these segments is making the entire male and female reproductive structures. So every one of these segments is a little hermaphrodite. And then what these worms can do is that they can bend over on themselves, or two tapeworms can come together and then exchange sperm from one segment to another and fertilize the eggs that are made in the ovaries there. And so every single segment is now creating hundreds of eggs, and there are thousands of segments. And then in the case of the rat tapeworm, what it does is it just pinches off the posterior, the end part of the body, to let go of those eggs so that they can go out in the rat poop, and that needs to get eaten by the intermediate host so that the larval tapeworms can now develop in that host. So in our case, a beetle can come along and eat this infected rat poop, and then they now become the host for the larval stage. The larvae develop in the beetle, and the rat can eat the beetle, and then the life cycle continues. But despite the fact that the worm is shedding parts of its body, and it has to shed its body in order to continue its life cycle, it does not just get shorter and die, it can continuously regenerate its body.
Because of those stem cells.
Because of those stem cells.
So basically 99% of the length of the tapeworm is reproductive.
Yes.
That explains why they're so good at it.
Exactly. That's amazing. So speaking of tapeworms, the tapeworm diet was fashionable way back in Victorian times.
Indeed.
But I hear it's still with us in some ways.
I do not endorse the tapeworm diet.
Well, we can talk about that for a second. But can you talk about what happens when a human is infected with a tapeworm?
Absolutely. So tapeworms are the stuff of nightmares, I'm sure, in people's imaginings. No one wants to think about a giant worm in their intestine. And if you were so unfortunate to have a tapeworm in your intestine, the truth is most of the time it will be asymptomatic. And the symptoms that you might experience are the ones that you would expect: intestinal discomfort, maybe some diarrhea, and vomiting. As a general rule of thumb, the bigger the tapeworm, the worse it's going to be, because the tapeworm is trying to eat your food. And so they might scavenge for things that are already limiting in our diet. The fish tapeworms, for example, they can get really, really large, and they tend to deplete from their human host vitamin B12, which can result in pernicious anemia or other immune and nerve health problems for the patient. But like I said, generally, the tapeworm wants you to survive and wants you to continue feeding it. So it has co-evolved with its host in the intestinal stage. The problem becomes when we become the hosts of the larval stages of tapeworms. And most of the time, in fact, it's possible that it's always an accident, that the larvae do not want to be in us. They are supposed to be in a different host system. And then the problem occurs because these larvae, they can take up many different forms. But in most cases, when humans are infected with larval tapeworms, they form these fluid-filled cysts. And they're not unlike tumors. They're going to infiltrate an organ system and cause all kinds of problems. And depending on the species, there's typically an organ system that is preferred by that species. And it might be the lungs or it might be the liver. In the case of the pork tapeworm, it has a propensity to be found in neural tissue, like in the eyes and even in the central nervous system, including the brain. So you can see how this becomes really severe. So the disease that is caused by the pork tapeworm when we are infected with the larval cysts is called neurocysticercosis. And it is actually one of, if not the leading cause of adult onset epilepsy. So people with these cysts, you can just imagine, it's so horrible. I've seen pictures of brain slices after autopsy, and these cysts are just infiltrating all the regions of the brain. And sometimes they need to be surgically removed. There's no other way to get rid of them. They can cause intracranial hypertension, seizures, blindness. In less severe cases, there still can be effects like learning disabilities that are caused by these tapeworm cysts. But I want to reiterate, you cannot actually get neurocysticercosis from eating undercooked pork. If you had done that and that pig was infected with a tapeworm, then you would get the adult tapeworm in your intestine. So now the human being becomes a carrier of this disease, and they're shedding the tapeworm eggs in their feces. So if this human doesn't have good hygiene practices and they don't wash their hands properly and prepare food, then they can contaminate the food with these tapeworm eggs that can be ingested by someone else. Or if the sanitary conditions are not so good and the sewage that is contaminated with these tapeworm eggs gets on the crops, that is another way that humans can become affected. So I don't know if this is more comforting or not, but you can be a vegetarian and still get neurocysticercosis because it is not from eating undercooked pork that you get the disease.
So I have a book in my office, New Guinea Tapeworms and Jewish Grandmothers. Have you read that?
I have. A long time ago, though. I need to revisit that one.
It's fun. It's just a lot of little vignettes about parasitology. We'll put a link to it on the show page. So,
do you want to give the punchline in relation to our conversation, or would you prefer that our readers check it out for themselves?
I'll let them check it out. Keep them interested. So back to the tapeworm diet, which I had to look that up, and I wasn't surprised to find references for Victorian times when it was probably a really big thing, but it's still with us. And what's the whole idea behind that?
Well, the idea is that if there's this big parasite that needs to feed on your diet, then it's making you thinner because it is absorbing all of the calories that are not going into you. But there's really no good evidence that this is true. And as I mentioned, the tapeworms themselves, they're not harmless. And the things that they need, are the same things that we need. But whatever we eat in abundance, they don't need as much. Whatever we eat that is limiting, they need more of. So they're actually depleting us of really essential nutrients, especially our vitamins, our amino acids, all of these things that are required for general human health. So yes, the tapeworm is eating your food, but not just in a way that'll make you thinner, rather a way that would make you less healthy. So it's not a good idea. Also, you have no idea what you're eating when someone gives you a tapeworm. It is not that easy to identify exactly what species of tapeworm, or if it is even a tapeworm that you have purchased. And this has definitely happened where people buy a tapeworm cyst off the internet and they think that this is going to give them a harmless worm in their intestine, and it is not.
That's
I don't even know what to say here. The lengths that people will go to for this quick fix is just unbelievable.
That's a horror movie right there.
Yeah. I'm just picturing in my head, I know it doesn't look this way, but a plate of spaghetti, that's actually worms that someone's eating.
It actually does look that way. Well, not if someone is trying a tapeworm diet.
Right.
But yeah, we do harvest tapeworms and have many large tapeworms in a large petri dish, and I have often reflected that it does in fact look like a bowl of fettuccine.
Little marinara sauce.
Garlic bread.
Yeah. Okay, well, our guest today is Dr. Tania Rozario from the Department of Genetics and the Center for Tropical and Emerging Global Diseases. Tania, thanks so much for being with us.
Thank you for having me. This was really fun.
Links to additional information about Dr. Rozario's research can be found on our Instagram account, which is at PPPPodcastUGA.
Thank you for tuning in today. Please rate the podcast. I'm thinking 5 stars sound good. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.